Hi, everyone. My name is Murat. I'm the CEO of Antiverse. Today, I would like to talk a little bit about the application of AI in the drug discovery and engineering. Now, when you think about it, before really getting into the details of Antiverse and what we do, I would like to paint a picture of how drug discovery is happening today. If you look at numbers coming from the industry, you would say that the cost of a drug coming from an idea to the market takes cost about $1 billion, $2 billion or more, including all the failures. And in terms of the timeline, it can be 10 years or more. So it is a very inefficient process. It's begging for some kind of innovation and transformation. And the belief and the conviction of many groups, including Antiverse, that AI can make a big impact to scale and improve that process. There are many companies working on what people call small molecules like aspirin and so on that can attack things between the cells, on the surface of the cells, inside the cells. And then you have these big molecules like antibodies that are very specific at what they bind, but they can only interact with things that are outside of the cell. So we are in the big molecule space. The problem with the big molecule space is there are less data, right? And that's one of the major problems when you try to apply an AI algorithm into this field. Now, within that space, you can again think of some people trying to design novel antibodies, and some of them are optimizing those antibodies for better characteristics. We are in the design space. We are creating this de novo antibodies against various different diseases. And again, within that space, you can see there are some AI companies, pure in silico. They do their work only computationally, and that's that. And there are other companies that incorporate a wet lab element. So we are one of those. Now I will go into a little bit more detail on what we do, but as a company, Antiverse, we founded the company in 2017 in an accelerator program in London called Deep Science Ventures. The idea was to rationally design an antibody, a drug, rather than trying to discover by trying, randomly billions and billions of different molecules and brute forcing your way through. Since then, we raised about $2.5 million. We are a team of 10. We work with big pharmaceutical companies and some biotechs and so on. We are mostly based in UK, but some of our people are in Turkey, and a few of them are in the US. Now, the problem we are really going after, and if you think about it, AI pitches usually can fall into two main sections. One of them, you say, with AI, we can now achieve something that wasn't achievable before. Or you can say that we are making an improvement to the existing processes, that we are 10 times faster, 5 times better, and so on. So in the pharmaceutical field, if you are in the second part, that you make a process better, you are a CRO, a contract research organization offering a fee for service and then making improvements and make them more competitive. We focus on the first part. We are offering something that wasn't possible before. So there are, I'm not sure the expertise level of the all audience. I'll try to explain a few of the concepts. We focus on receptors associated with disease attached on the cell surface. These are GPCRs, ion channels, and some other receptors. There are hundreds of them associated with tons of different diseases. But as a whole industry, we only managed to find or generate 2 antibodies against them. So something has to change. Why is it that bad? There are 2 problems. I call them as a forest problem and an iceberg problem. So first of all, these receptors that you want to modulate, they are embedded among a forest of other receptors. So finding them is very difficult. And even if you find them, then there's the iceberg problem. most of that receptor is within the cell, so you cannot connect to that. And there's a tiny little bit outside that you really have to focus on. Our solution has two parts that corresponding to those problems. So we created new cells in our lab that has more of that receptor. They are still hidden among that, forest, but because we have 1,000 times more copy, it increases our chances of hitting those targets. And the second part is where AI comes into the picture. Because there's such a tiny bit, you need to design antibodies predicted to bind to that particular target. So we were focusing on this aspect about now 6 years, and we made a lot of progress, which I will get to in a minute. So for the first part, I'll skip this slide very quickly, but we work with Gersi Hussain. He was the former chief scientific officer of Teranix to develop hundreds of such cell lines. And we have access to about 140 of them. And in our tissue culture lab, we generate more of such cell lines. Now, for the AI machine learning part, we found out that we need a lot of data. So we collected what is available online, and then we started to work with pharmaceutical companies and get their data in, but still it wasn't enough. So we set up a lab process where the idea, the objective is not antibody discovery, but data generation. So we took off-the-shelf targets, and then we designed AI-based antibody libraries. We screened those antibody libraries against these targets, and now found what binds to what and at what strength level, and then put that into a database. Then we trained new ML models on that data, which infers patterns of, okay, this kind of target and this type of antibody, they work well together. Now, once that information is captured into the machine learning algorithm, we point that algorithm to new targets and then make predictions of novel antibodies that would possibly bind to that target. Our main KPI that we have been tracking for a long time was how many antibodies we need to generate, and you can think of it as potential treatment, how many antibodies we need to generate to find one binder against a given target. And that number was awfully bad. Billions and billions and billions. It was beyond the realm of synthesizing and testing them. But progress after progress, we brought that to first, sub 100,000s level, and eventually 1 in 18,000. What it means, now you can spend about $10,000 to synthesize about 100K antibodies as a form of library and screen those libraries against this new target and expect to find 5 good antibodies. Our models so far are sequence-based. So sequence of a target to the sequence of an antibody. And we managed to push the accuracy to 1 in 18,000, but we want to go beyond that. We want to come into 1 in hundreds. And if we can do that, we can shave off another 3 months for our development process. However, that kind of accuracy requires structural information, which doesn't exist. So what you can do, then you can try to generate that 3D structures from the blueprint, from the sequence of these antibodies and targets. Now, I'm sure you have heard of AlphaFold. AlphaFold can, for example, make an antibody prediction. It takes about 15 minutes per structure, and it is very, very low throughput for our work. So we had to build our own protein structure prediction machine learning algorithms. And now we can predict 16 structures per second, opposed to 1 structure in 15 minutes. And with that, we can screen now millions of antibodies in a day. And the accuracy is not bad. If you're familiar with the field, sub-angstrom accuracy is an experimental accuracy. So you do the same experimental test in 2 different labs, and you would expect to get to a sub-angstrom. There will be some deviation. So we reach almost that type of accuracy when we make our antibody structure predictions. And there are a couple of examples on the right-hand side. Blue is the ground truth, and our prediction is in green. So how does it work for pharma? What do? We know that they have these difficult targets that they are not making any progress with. So we go to them and we talk to them. We say, look, you probably have some inactive projects that are associated with cancer, heart, lung conditions, addiction, inflammation, you name it. We know that you spend a lot of time and you spend a lot of money. We can give it another shot. Now there's a new drug discovery development platform in town. So you tell us what the target is, and what we will do in 6 months from now, we will give you a functional antibody. So it will bind to your target, but not that it will only bind to your target, but it will also create some type of function, upregulating, downregulating, whatever is necessary for that disease. We do most of that work internally, but we outsource certain bits and pieces. So this is a work that we have done with a big pharma. And if you look at this graph, so all of these bits, every single dot here is an antibody. This is the output of a clustering algorithm. It has been done in a 20-dimensional space, but when you want to report it, you reduce it to 2D, or if you count the colors in a 3D format. So these are all antibodies that is expected to a certain degree interact with a target. So you cannot just simply go and test all of those antibodies. So what we do for the pharma is saying that, look, there seems to be grouping of antibodies, and we pick a representative antibody from each group and we give them that final list to test. And imagine that an antibody from the salmon red color group turn out to be functional, but maybe missing some other characteristics, like not binding strongly enough, or solubility or developability issues. We go back to that group and can pick maybe another 96 antibodies for them to test. This is another work we had done with a pharma where we showed that. I'll just run if you focus on maybe just this row, we show that we can find 87 different antibodies where they could only find 38 of them. And out of that 87, there were a couple of them are cross-reactive. What it means that it interacts with the target of your choice, but it also interacts with a similar target in an animal model. So you can use the same antibody to do your animal testing and bring it into the clinic without any change. So it's something desired. And the accuracy of our predictions of such antibodies were 94%. In another example, we discovered antibodies from our libraries, which are designed by machine learning again. And we found antibodies competitive with Keytruda. Keytruda is one of the biggest antibodies used in all sorts of things. It was one of the PD-1 antibodies. These drugs, just to give you an example also, can be making billions in revenue in a year. So it's a big market. There's a huge need. So many people are going after it, but there's not a lot of success. Other than working with pharmaceutical companies, what I want to do, we want to help as many people as we can. That's why we started Antiverse. So our idea is not do a little bit of work and then sell it to a big pharma or a big contract research organization. We want to bring this company all the way to the market and runs hundreds of programs where a similar company can run only a handful. Because we reduce the cost of running a campaign so much. So now in our pipeline, we started with 6 different programs. They are all cancer related, but they have secondary, tertiary indications. And for PAR1, we are expecting our first functional output, and hopefully the results will come in fine in 7th of November, and then we will push a few of those candidates into animal studies. This is another example. So this is a GPCR. This is an antibody. If you see my cursor, and this is this tiny little bit you try to bind to. So just to a little bit express the difficulty of the task. And these are the things that we are designing, that final interaction by using AI. I'll skip this one very quickly, but against the PAR1, the GPCR that you guys just have seen, we designed multiple antibodies, and 17 of them showed some level of binding, and now we are testing them for further function. So companies like ours in this space, they work in two formats, like I mentioned before. Some of them are fee-for-service, and others, they are like us, pushing for partnerships. If you create a lot of value, then you can leverage that value to get into contracts where you can ask for milestones and royalties. So that's our business model. That's what we go with here. They tell us what the target is. We build a cell that expresses that target million times on it. And then we designed, by using AI, a target specific library. As far as I'm aware, nobody has done that for the target, at least in the antibody space. Then we screen those antibodies against the target, find something, and then test it for function. And the numbers, so upfront fees for more established companies range from $1 million to 10 million per program. For us, it's more on the 100K range. But then the milestones that you collect, it's around 30 million in total, and you expect to get 150 to 500 million from the royalties. And if you look at Atomwise, for example, from US, they work in the small molecule space, and they signed 5 deals with big pharma in 2019 alone, ranging from $500 million to $2.5 billion. So that's the kind of business model, that's the kind of potential there commercially for this type of approach. Maybe I'll briefly talk about my background and my co-founder's background. And I'm really hoping that we will attract more programmers, engineers into the space. So my background is in electrical engineering and computer science. I did programming for many years. Then when my father got diagnosed with lung cancer, it piqued my interest to this field. I stayed in finance for a while, and I saw how many biologists are in finance, and it just blew my mind. And I said, okay, maybe if people can move from life science to finance, maybe I can move from finance to life sciences. So that's what I've done. And I wanted to do something with my skills that make the life of many patients better, whether it is cancer, whether it's addiction, inflammation, heart, lung conditions, and all others. That's where I'm coming from. My co-founder, with my background, I could generate a lot of data. I can automate hardware, software, but I needed somebody smarter than me to look at that data and build a mathematical model. So I met my co-founder, Ben Holland. He's an engineering science degree from Oxford. He's been all around the world, multiple patents to his name. So we did a prototype in 3 months, and then had a Dragon's Den type of setup, and got our initial funding in 2017. That was 70K. And since then, we kept repeating that cycle. I think I'm going to follow suit and finish a little bit early. I would like to thank you to all of you for listening to me. Thank you very much.